Every living body moves, breathes, responds, and holds itself together in ways so constant that they almost go unnoticed. Until one day, all of it begins to fade. What happens in the final minutes? What does the brain experience as oxygen falls away? When does consciousness end? And what can science actually say with confidence, without myth and without exaggeration? Tonight, this documentary follows death from the outside in, through the body, the brain, the cells, and the final boundary of what science can truly know. If this is the kind of thoughtful documentary content worth making, please like, subscribe, and support the channel. Death first appears in the world around a person as a change in relation. A living human being is never entirely still, even in sleep. The chest rises and falls, the throat shifts with swallowing, the face makes small adjustments, the fingers curl and release, the eyes move beneath their lids, warmth spreads outward from skin into sheets, clothing, air, and the hands of anyone nearby. Breath leaves a trace in cool light. The body answers its surroundings in countless quiet ways. It responds to touch, to sound, to pain, to pressure, to the simple pull of gravity. When death arrives, the first region science can describe is not hidden deep inside tissue or cell. It is this outer field of visible connection, the place where life shows itself in motion, heat and response. In that outer field, the difference is often plain before it is fully understood. A room that held a living person begins to hold a body that no longer participates in the room in the same way. The body remains present in shape, weight, and familiar features. Yet something measurable has changed. The surface of the skin may lose its usual colour as blood flow weakens and then stops. The muscles no longer make the tiny corrections that keep the head balanced or the mouth softly closed. Eyelids may stay partly open or fully shut depending on circumstance, but they no longer blink. The face, which once carries expression even in rest, becomes less active, less variable, less involved with the passing moment. Observers often describe this change in simple terms. The person seems absent, even while the body remains entirely there. Science approaches that impression by looking at function rather than feeling. A living body continuously exchanges matter and energy with its environment. It takes in oxygen, gives off carbon dioxide, releases heat, moves fluid, maintains electrical signals, and preserves the delicate differences between inside and outside that every cell requires. At the most outward level, these inner processes create signs that other people can see and feel. When the signs vanish together, the absence is not imagined. It reflects a real loss of organized activity. The stillness of death is therefore different from the stillness of sleep, sedation, or unconsciousness. In those conditions, the body still works as a coordinated whole. Breathing continues, even if slowly. Circulation continues. The brain remains active enough to regulate temperature, pressure, and many essential rhythms. A sleeping person can usually be roused. A sedated person may need time or stronger stimulation, but the body still shows signs of ongoing control. In death, that coordination is ending or has ended. The body no longer maintains the outward patterns that living systems sustain without pause. One of the clearest outward signs is the loss of responsiveness. Responsiveness means the ability of the nervous system and muscles to react to the world. A hand touched by cold may withdraw. A loud sound may trigger a blink or a change in breathing. Pain may cause a grimace, a movement, or a vocal sound. Even a person in severe illness usually preserves at least some small response until very near the end. As death approaches, these signs often fade. Reaction becomes delayed, then weak, then absent. The eyes may no longer track movement. The face may no longer tighten against discomfort. The limbs may rest heavily, no longer resisting repositioning. This loss has a physical basis. The systems that detect the world, carry signals, and produce action are beginning to fail. From the outside, however, it appears simply as a growing silence. Breathing is part of this outer silence as well, though in the earlier stage it may still be present. What matters first is not the hidden chemistry of the lungs, but the visible pattern of air moving in and out. In life, breathing creates a rhythm that others often notice without thinking. Fabric shifts, ribs lift, nostrils move. The throat makes subtle adjustments. Near death, that rhythm may become irregular, shallow, or widely spaced. At the outer edge of observation, the important fact is that the once steady conversation between body and air is changing. When the rhythm weakens, the room itself seems to change with it, because a human presence is partly defined by breath entering and leaving shared space. Warmth also belongs to this outer region. A living body produces heat through metabolism, the sum of chemical reactions that keep cells alive. Blood carries that heat outward, and skin releases it into the surrounding air. A hand placed on a living forearm feels not only temperature, but active warmth, a sense of heat being renewed from within. After death, this process no longer continues. At first, the body may still feel warm, because heat does not disappear at once, yet the quality changes. The warmth is no longer maintained. It begins a gradual decline toward the temperature of the surroundings. In ordinary experience, this is one of the earliest physical differences another person may notice, especially in the hands, feet, and face. Posture changes too. Life requires constant low-level muscle activity called tone, the slight tension that helps maintain shape and position. A living person at rest still holds the jaw, neck, shoulders, and hands with this faint internal support. As death nears and passes, muscle tone diminishes. The mouth may fall open, the limbs may become heavy, the head may turn more freely. The body seems less like an actor within the environment and more like an object within it. This shift is not complete at once, and later stages bring other changes, but the early outer impression is one of release from active control. Science also notes the role of perception in this first region. The surrounding people do not directly observe cells losing energy or neurons losing coordinated firing. They observe stillness, silence, pallor, cooling, and lack of response. These outward signs matter because they are how humans have always recognized death before instruments, hospitals, or laboratory knowledge. Modern medicine adds tools such as stethoscopes, monitors, and brain recordings. But the first encounter remains sensory. The body no longer behaves as a living body behaves. The ancient observation and the modern explanation meet at this surface. Even here, science is careful. A body that appears lifeless is not always permanently dead at first glance. Severe cold, poisoning, trauma, certain drugs, or rare medical states can reduce visible signs of life so much that trained examination is needed. This caution is one reason medicine defines death through specific criteria rather than impression alone. Still, the visible transformation remains meaningful. It marks the moment when the outer world ceases to receive ordinary signs of living organization. So the scientific answer begins not with mystery, but with withdrawal from contact. The world around the dying person receives fewer signals, then none. Movement ceases, response fades, warmth stops renewing itself, breath weakens into still air, the body remains, but its active exchange with the surroundings grows quiet. At this outermost level, death is the end of visible participation in the immediate world, the point at which a human organism no longer shows the signs by which life makes itself known on its surface. In the final shared moments, the outward stillness of the body is not yet complete, but it is becoming established. The surrounding space continues to register small, fading signals that life is present, though the signals no longer follow the steady patterns seen in health. The body still participates in the environment, but its participation is irregular, weakened, and often fragmented. Science observes this stage through measurable changes in breathing, skin, and movement, all of which reflect deeper processes that are beginning to fail. Breathing, which once follows a smooth and continuous rhythm, may shift into uneven patterns. The chest may rise and fall with long pauses between movements, or with clusters of breath separated by silence. These changes are linked to the brain's control over respiration, which becomes less stable as oxygen levels fluctuate and neural coordination weakens. From the outside, the important detail is the interruption of predictability. The regular exchange between body and air begins to break into segments, as if the system guiding it is losing coherence. The sound of breathing may also change. Air passing through the throat can produce soft, irregular noises when muscles no longer manage airflow with precision. Fluids may accumulate in the airways because swallowing reflexes weaken, leading to a low, intermittent sound with each breath. These effects are well documented in clinical observation and arise from the gradual loss of muscular control rather than any sudden event. The body is still attempting to breathe, but the coordination required for quiet, efficient airflow is diminishing. Skin provides another outward signal. Color may shift as circulation becomes less effective. In a healthy state, blood flow delivers oxygen to tissues and maintains consistent tone across the surface. Near death, this distribution becomes uneven. Areas of the skin may appear pale or slightly bluish, particularly at the extremities such as fingers, toes, and lips. This change reflects reduced oxygen delivery and slower blood movement. It is not a single moment but a progression, where the body surface gradually loses the balanced appearance created by active circulation. Temperature at the surface can vary during this period. While the body still produces some heat, reduced blood flow to the outer regions may make the skin feel cooler in certain areas. Hands and feet often show this first, as circulation prioritizes central organs. This redistribution is part of the body's attempt to preserve essential function, even as overall capacity declines. Observers may notice a contrast between warmer central regions and cooler extremities, indicating that internal systems are no longer maintaining uniform conditions. Muscle activity continues to change. Movements that were once voluntary may become limited or absent. At times, small, involuntary motions can occur, such as slight twitches or shifts in position. These are not organized actions, but rather residual activity within the nervous system as it loses control. Muscle tone decreases further, and the body becomes increasingly relaxed in a passive sense. Limbs may rest in positions determined more by gravity than by internal support. Awareness, though not directly visible, leaves traces in outward behavior. A person nearing death may no longer respond to voices, touch, or changes in the environment. In earlier stages, there may still be brief moments of reaction, such as a slight movement of the eyes or a change in breathing when stimulated. As the process continues, these responses become less frequent and eventually cease. Science understands this as a result of declining brain function, particularly in regions responsible for integrating sensory input and producing coordinated output. The eyes themselves may show changes that are observable from the outside. The gaze may become unfocused or fixed, no longer tracking movement or adjusting to light in the usual way. Pupils, which normally constrict or expand in response to brightness, may react more slowly or not at all as neural control diminishes. Eyelids may remain partially open without blinking, reflecting the loss of reflexive muscle action. These signs are subtle but consistent with the broader pattern of decreasing responsiveness. Circulation, though still present at this stage, becomes weaker and less efficient. The pulse may be difficult to detect without careful examination. Blood pressure tends to fall as the heart struggles to maintain forceful contractions. While these details require instruments to measure precisely, their effects appear externally through the changes in skin color, temperature, and overall vitality. The body's outward appearance becomes less dynamic, reflecting the reduced flow of blood within. The environment around the person remains unchanged in its physical structure, yet its relationship with the body is altered. Air continues to move, light continues to fall, and objects remain in place. But the body no longer interacts with these elements in a full and responsive way. The exchange becomes one-sided. The environment acts upon the body, but the body does not answer with the same degree of activity. This imbalance is a defining feature of the final shared moments. Medical science approaches this stage with careful observation and defined criteria. Clinicians look for patterns rather than single signs, understanding that each change contributes to a broader picture of decline. No single outward sign confirms death at this point, but the combination of irregular breathing, reduced responsiveness, altered skin tone, and weakening circulation indicates that the body is approaching a critical threshold. These observations guide decisions about care, but they also reflect fundamental biological processes that can be studied and described. It remains important to distinguish this stage from the moment when all vital functions cease. In the final shared moments, the body is still active in limited ways. Breathing, though irregular, continues. The heart still moves blood, though with reduced force. Cells still receive some oxygen and nutrients. The processes of life are present but unstable. Science describes this as a transition rather than an end point, where systems are failing but not yet entirely stopped. Within this outward region, uncertainty is minimal regarding what can be measured. Observations consistently show that these patterns occur across different individuals and conditions. What remains uncertain is the exact internal experience, as direct measurement of consciousness is limited. However, the outward signs provide reliable evidence of declining physiological function. The body is no longer maintaining the complex coordination required for sustained life. Thus, the final shared moments are defined by fading interaction. The body continues to exist within its surroundings, but its ability to respond, regulate, and sustain visible life diminishes steadily. Breathing loses rhythm, skin loses balanced color, movement becomes rare, and awareness no longer expresses itself outwardly. These changes mark the closing of the boundary between life and death, at the level where it can be seen and felt by others. As the body moves beyond the last outward signs shared with others, the scientific view turns to the broad internal systems that keep a human organism alive from moment to moment. In this region, death is not yet a condition of still organs and silent tissue, but a process of failing support, breath and blood, which normally work together without pause, begin to lose their ability to sustain the body's balance. Air may still move into the lungs, the heart may still produce some circulation, yet these actions no longer provide what the organs need in a stable and continuous way. The central fact of this stage is not complete absence, but insufficiency. The body's great transport systems remain present while no longer meeting the demands of living tissue. Under ordinary conditions, breathing and circulation form a linked chain. The lungs bring oxygen into contact with blood through tiny air sacs called alveoli, which are small sacs at the ends of the airways where gases pass between air and blood. Oxygen moves across a thin barrier into the bloodstream, while carbon dioxide moves out to be exhaled. The heart then pushes oxygen-rich blood through arteries to the brain, kidneys, liver, muscles, and every other organ. Veins return blood carrying carbon dioxide and chemical waste. This continuous movement preserves temperature, pressure, acidity, salt balance, and cellular energy production. Life depends on the reliability of that movement. There is very little reserve. A human body can tolerate interruption only briefly before function begins to fail. Near death, this support weakens in ways that can begin gradually or happen suddenly, depending on the cause. In many forms of dying, breathing becomes less effective before it stops altogether. Air may still enter the lungs, but not deeply enough or regularly enough. The muscles of breathing, especially the diaphragm and chest muscles, may tire or lose coordinated control. Fluid may collect in the lungs. Airways may narrow. The brain centers that regulate breathing may no longer respond normally to rising carbon dioxide. The result is that the body draws in less oxygen and removes carbon dioxide less efficiently. The exchange does not end at once, but it becomes poor. This decline has immediate consequences for blood chemistry. Oxygen levels begin to fall. Carbon dioxide often rises. As carbon dioxide accumulates, the blood becomes more acidic. Acidity in this context means a shift in chemical balance that affects how proteins, enzymes, and cell membranes work. The body can compensate for modest changes for a time, but near death these compensations weaken. Breathing may become faster for a period as the body attempts to correct the imbalance. Later, as control systems fail, the pattern may slow and fragment instead. From the scientific perspective, the lungs are no longer serving as a reliable gateway between the body and the air around it. Circulation is failing at the same time. The heart may still beat, but a heartbeat alone does not guarantee adequate blood flow. Effective circulation depends on force, rhythm, vessel tone, blood volume, and the capacity of the heart muscle itself. Near death, blood pressure often drops because the heart cannot contract strongly enough, because blood vessels lose coordinated control, because blood volume is reduced, or because severe illness disrupts several of these at once. Blood pressure is the force blood exerts on vessel walls, and it is essential for pushing blood through organs that depend on constant supply. As pressure falls, the body begins to favor the organs most necessary for immediate survival, especially the brain and heart, while reducing flow to skin, limbs, intestines, and kidneys. This redistribution explains many outward changes already seen, but inside the body it has deeper effects. The kidneys receive less blood and produce less urine. The digestive system slows because it is no longer a priority for survival. The liver, which normally processes nutrients, toxins, and waste products, loses efficiency. Muscles receive less oxygen and become weaker. The skin cools because warm blood is no longer reaching it in the same quantity. The body is not choosing freely among its organs. but following automatic survival mechanisms that narrow support to the most essential regions. These mechanisms can delay complete collapse for a short time, yet they also reveal how close the organism is to losing system-wide coordination. The blood itself changes as circulation slows. In life, blood is a fast-moving transport medium. It delivers oxygen, glucose, hormones, immune cells, and clotting factors while carrying away waste. When movement becomes sluggish, delivery and removal both suffer. Tissues that already receive limited flow become more deprived. Waste products accumulate in local areas. Small vessels may no longer fill evenly. Cells that line blood vessels can become damaged by low oxygen and inflammation, making circulation even less efficient. In some dying processes, especially those associated with infection or severe trauma, the normal balance between clotting and free flow also breaks down, further reducing the body's ability to move blood where it is needed. The brain is especially sensitive to these changes, but in this section the focus remains on the failing support itself. What matters here is that breath and blood are not separate events. A weak breath means less oxygen enters the blood. A weak circulation means what oxygen remains does not reach tissues effectively. If carbon dioxide rises and acidity increases, heart muscle and blood vessels work less well, which makes circulation worse. This in turn further reduces oxygen delivery to the brain areas that regulate breathing and heart function. The decline becomes circular. Each weakness intensifies the others. The body enters a state where compensation no longer restores balance, but only slows its loss. Even at this stage, the process is not always smooth. There may be brief fluctuations that seem like improvement. A few stronger breaths may appear after a period of shallow breathing. The pulse may momentarily feel fuller. Skin color may shift slightly as blood flow changes. These variations do not necessarily mean recovery. In many cases, they reflect unstable control systems making short-lived adjustments in a body that can no longer sustain them. Scientific observation shows that dying often includes irregularity because biological systems break down unevenly. Some tissues retain function longer than others. Some reflexes persist after broader coordination is gone. The overall direction, however, remains one of declining support. Time in this region is measured in minutes to hours in many cases, though the exact pace depends strongly on cause. Sudden injury, severe heart failure, advanced infection, massive blood loss, drug overdose, and neurological catastrophe can all bring the body to this stage by different routes. Yet the shared pattern is clear. Oxygen delivery falls below need. Carbon dioxide removal becomes inadequate. Blood pressure can no longer sustain organ perfusion, meaning the flow of blood through tissues. The organs continue to exist as structures, but the flowing support that keeps them alive is diminishing past the point of stability. This is the broad internal threshold of dying. The body is still a whole organism in form, but no longer in performance. Breath is becoming too shallow, too irregular, or too ineffective to maintain normal gas exchange. Blood is moving with too little force and too little reliability to nourish the organs. The lungs and heart, which sustain all other tissues through exchange and transport, are no longer able to preserve the environment that living cells require. Death in this region is the slow withdrawal of support from the entire body, the failure of air and blood to continue the work they have carried from the first breath onward. At the next inward layer, the failing movement of blood reaches its decisive limit. The heart, which has continued to contract with declining strength or irregular timing, stops producing organized circulation. This event is called cardiac arrest. In simple terms, cardiac arrest means the heart can no longer pump blood in a way that sustains the body. it does not always mean the heart has become motionless in the same instant. It means the heart's activity is no longer effective as a pump. The difference matters because life depends not on movement alone, but on coordinated movement that sends blood forward through the vessels. In a healthy body, the heart works as a muscular chamber system driven by electrical signals. These signals begin in specialized cells, spread through the upper chambers, pause briefly, and then move through the lower chambers in a sequence that produces strong synchronized contraction. That timing allows blood to fill, then be ejected into the lungs and the rest of the body. Each beat is not merely a squeeze, but a patterned event shaped by structure, electricity, pressure, and valve function. The heart must repeat this with remarkable reliability, day and night, over many years. When death approaches, that order can weaken gradually or collapse suddenly. The key transition in this section is the end of organized forward flow. Cardiac arrest occurs in several ways. In some cases, the heart's electrical system becomes chaotic. Instead of a coordinated wave of activation, the muscle fibers contract in a fast, disordered pattern known as ventricular fibrillation. In that state, the heart quivers rather than pumps. Blood is not propelled effectively, even though electrical activity continues. In other cases, the heart becomes so electrically and mechanically weak that it slows to a stop. This may happen after prolonged illness, severe oxygen deprivation, major blood loss, or failure of the heart muscle itself. There is also pulseless electrical activity, in which electrical signals may still appear, but the muscle does not generate a meaningful pulse. Science distinguishes among these patterns because they differ in mechanism, but the shared outcome is the same. Circulation fails. The moment circulation ceases, the entire body enters a new condition. Until now, organs have been supported poorly, yet still supported. With cardiac arrest, that support is no longer merely insufficient. It is absent as a coordinated system. Blood pressure falls towards zero because there is no force maintaining it. Arteries no longer carry pulses to distant tissues. Veins no longer return blood in a living circuit shaped by the heartbeat. Blood remains in the vessels, but it is no longer in rapid movement. This is why the pulse disappears. A pulse is not simply blood existing within the body. It is the transmitted wave created by each effective heartbeat. When the pumping action ends, that wave vanishes. This boundary has great importance in medicine because it marks a major point of clinical death. Clinical death refers to the state in which breathing and circulation have ceased. It is not yet the same as complete biological breakdown of every cell. Some tissues remain alive briefly after circulation stops. Some may still be recoverable if blood flow is restored quickly enough. This is the basis for resuscitation. The attempt to re-establish heartbeat and breathing before irreversible injury spreads through the brain and other organs. But without prompt restoration, the loss of circulation begins a rapid cascade. The heart stopping is therefore both an event and a gateway. It ends system-wide delivery in the present and begins system-wide injury in the minutes that follow. The heart itself is changed by this event. A living heart muscle depends on its own blood supply, delivered through the coronary arteries. When organized pumping stops, these vessels no longer receive normal flow. The heart becomes both the cause of circulatory failure and one of its victims. Without oxygen and fuel, heart muscle cells begin to lose the chemical gradients that allow contraction. Calcium handling becomes abnormal. Energy stores fall. The organized electrical behavior that once drove each beat becomes harder to sustain. In a brief arrest with successful resuscitation, some of this can recover. In prolonged arrest, The damage deepens until the heart can no longer return to coordinated function. Elsewhere in the body, the absence of circulation has immediate physical meaning. Oxygen delivery stops, glucose delivery falls away, waste removal nearly ceases, heat is no longer actively distributed from core to surface. The body still contains blood, but blood without motion cannot serve its living purpose. Organs are not nourished by stored presence alone. They require continuous flow. This distinction is central to the scientific understanding of death. Life is not a fixed substance held inside the body. It is a process maintained by constant movement, exchange, and regulation. Cardiac arrest interrupts that movement at its core. The skin may change quickly after this point. Colour often becomes more pale because there is no longer active circulation near the surface. Warmth begins to withdraw from the extremities more decisively. The body may appear suddenly more distant from life, even before later post-mortem changes begin. Yet these are still surface reflections of the deeper event, which is the end of pump-driven circulation. At the level of the whole organism, the heart's stop creates stillness inside the vessels, stillness in pressure, and stillness in the supply lines on which every organ depends. From a scientific perspective, cardiac arrest is not always a single clean instant that can be sharply separated from the minutes before it. In some dying processes, the heart becomes weaker over time, with falling pressure and intermittent rhythms before organized output fully ceases. In sudden collapse, the shift may be abrupt. Instruments can mark the loss of pulse, the disappearance of measurable blood pressure, and the electrical pattern on a monitor. But the body experiences it as the end of effective transport. Whatever the route, the threshold is crossed when the heart no longer sustains circulation in a meaningful way. This is why medicine treats time after cardiac arrest with such urgency. The brain can endure only a short period without blood flow before severe injury begins. The heart muscle itself deteriorates. The chances of recovery fall with each passing minute unless cooling, immediate resuscitation, or other exceptional conditions delay damage. Scientific consensus is clear on this point. Human tissues, especially brain tissue, rely on continuous circulation and begin to fail rapidly when it stops. Cardiac arrest is therefore one of the clearest boundaries between unstable life and active dying. There is also an important difference between cardiac arrest and a heart attack, though the two can be related. A heart attack usually means loss of blood supply to part of the heart muscle, often due to a blocked coronary artery. Cardiac arrest means the heart stops pumping effectively. A heart attack can trigger cardiac arrest, but not all cardiac arrests are caused by heart attacks. This distinction helps define the event more precisely. The body dies not because the heart has suffered one particular kind of injury, but because the circulation that supports all tissues has come to an end. At this inner stage, death becomes a matter of halted transport. The body remains intact in outline, weight, and familiar form. But the central engine of forward blood flow is no longer working. The heart's contractions, if any remain, are disordered, too weak, or too brief to maintain life. Pressure collapses, pulse disappears, blood ceases to travel as a living stream. Cardiac arrest marks the point where the body loses its organized circulation, and with it the ability to sustain itself as a coordinated human organism. Once the heart stops providing organized circulation, the body's deepest dependence becomes plain. The brain loses fuel. This loss is both simple and severe. Brain tissue depends on a constant supply of oxygen and glucose, a simple sugar that cells use as a primary energy source. Unlike some other tissues, the brain stores very little of either in a form that can support prolonged activity. It works continuously, and it draws heavily on the blood for immediate delivery. The heart and lungs have already begun to fail as reliable support. Now, at this more inward level, the consequence reaches the organ that coordinates sensation, memory, movement, attention, and the body's own internal regulation. The brain is still present inside the skull, but the flow that sustains its work is falling away. In ordinary life, the brain receives a large share of the blood pumped by the heart. Though it makes up only a small fraction of the body's weight, it consumes a high proportion of the oxygen and glucose carried in the blood. Neurons, the specialized cells that send electrical signals, require this constant supply because their activity depends on maintaining tiny differences in charged particles across their membranes. These differences are not passive. They are actively maintained by molecular pumps that consume energy at every moment. When oxygen and glucose are available, cells produce the energy molecule called adenosine triphosphate, which powers these pumps and many other processes. When blood flow stops, that energy production begins to fail within minutes. The first change is not visible from outside the skull. It occurs in metabolism, the chemistry of life within the tissue. Oxygen is required for the most efficient form of cellular energy production. Glucose provides the raw material. Without circulation, oxygen delivery stops and glucose delivery stops with it. The brain must rely briefly on what remains locally available in blood vessels and cells. Those tiny reserves disappear quickly. Energy production falls. As it falls, neurons become less able to preserve the electrical conditions needed for normal signaling. The brain is not a solid block that shuts down all at once. It is a vast network of cells and connections, each with slightly different demands and vulnerabilities. Even so, the general direction is rapid. Fuel loss begins to silence function. This silence first affects the highest and most energy-demanding activities. The brain's outer regions, especially the cerebral cortex, support awareness, language, planning, memory, and the integrated sense of being a person in a place and time. These functions rely on widespread coordination across many regions. They are therefore especially sensitive to disruption. When blood flow declines below critical levels, organized thought becomes difficult or impossible. Attention fragments. Perception narrows. The ability to form new memories fails. In medical observation, a person may become confused, unresponsive, or unconscious as this process deepens. These outward states reflect the internal fact that large networks can no longer support coordinated activity. At the same time, deeper structures that regulate arousal and basic bodily control are also under strain. The brainstem, which helps regulate breathing, heart rate, swallowing, and alertness, is somewhat more resistant than the cortex in some circumstances, but it too depends on blood flow and oxygen. So do the thalamus, which helps relay sensory information, and other subcortical regions involved in movement and consciousness. The loss of fuel, therefore, does not affect only thought. It begins to compromise the very systems that hold wakefulness and bodily integration together. The organism loses not just awareness of the world, but the brain's capacity to maintain its own internal order. The mechanism of this failure is grounded in basic cell biology. Neurons maintain a difference in sodium, potassium, calcium, and other ions across their membranes. This ion balance allows them to generate and transmit electrical impulses. The pumps that sustain the balance require energy. When energy runs low, the gradients begin to collapse. Sodium enters cells more freely. Potassium leaks out. Water follows the shifting chemical balance, and cells may begin to swell. Electrical signaling becomes unstable. Some neurons stop firing in organized ways, others depolarize, meaning they lose the voltage difference that allows controlled activity. In living function, the brain depends not simply on electrical activity, but on patterned electrical activity. When fuel loss disrupts the pattern, function disappears even before all cells are structurally destroyed. Chemical imbalance also spreads through the tissue. Without oxygen, cells switch to less efficient forms of energy production, which generate lactic acid and contribute to rising acidity. This further disturbs enzymes and membranes. Neurotransmitters, the chemicals neurons use to communicate, may accumulate abnormally in the spaces between cells because normal uptake and recycling require energy. One important neurotransmitter in this context is glutamate, which normally helps excite neurons and support signaling. In excess, however, glutamate contributes to injury by overstimulating receptors and allowing too much calcium to enter cells. Calcium is essential in controlled amounts, but in excess it activates destructive pathways. Thus, the loss of fuel does not merely turn the brain off. It begins to convert normal signaling mechanisms into sources of damage. Different parts of the brain tolerate this process for different lengths of time. Some neurons are especially vulnerable, including certain cells in the hippocampus, a structure important for memory, and in parts of the cerebral cortex and cerebellum. Other cells may survive somewhat longer if blood flow is restored quickly. This uneven vulnerability is one reason recovery after cardiac arrest, when it occurs, can be incomplete and variable. But in the ongoing dying process, the main scientific point remains clear. The brain requires uninterrupted metabolic support, and once circulation fails, that support declines too fast for normal function to continue. Unconsciousness often develops early in this region because awareness depends on integrated activity across large networks. Even a short interruption in circulation can break that integration. The body may still retain some reflexes for a brief period, and isolated neurons may still fire. But the coordinated pattern necessary for ordinary consciousness is already failing. This distinction is important. The brain does not need to be completely inactive for awareness to disappear. It only needs to lose the organized, sustained activity that binds sensation, memory, and self into a coherent state. Temperature, previous illness, and cause of death can influence the pace. In rare cases of profound cooling, the brain's metabolic demand is lowered, and tissue can survive longer without normal circulation. This is one reason some cold water drownings or severe hypothermia cases allow recovery after unusually long intervals. But under typical human conditions, the brain's loss of fuel is swift and unforgiving. Scientific consensus supports this strongly. Brain tissue is among the least tolerant tissues in the body when deprived of oxygen and glucose. What can be measured in this stage is substantial. Doctors can observe loss of responsiveness, absence of purposeful movement, changes in pupil reaction, and later changes in brain electrical activity. What remains less direct is the exact internal experience during the first moments of fuel loss. Some people who are resuscitated later report perceptions or memories associated with near-death states, but such reports do not change the known physiology. Observations indicate that as blood flow falls, the conditions required for normal consciousness deteriorate rapidly. It remains uncertain exactly how subjective experience maps onto those final moments in every case. At this inward layer, death is the deprivation of the brain's immediate necessities. Oxygen no longer arrives in adequate amounts. Glucose is no longer delivered. Energy production weakens. Ion gradients decay. Signaling loses its order. The brain, which governs awareness and coordinates the body, begins to fail because its fuel is gone. The loss is not symbolic. It is physical, chemical, and measurable. The organ that depends most intensely on constant support is now entering the state where function can no longer be sustained. As the brain loses its steady supply of oxygen and glucose, the changes in awareness become more complete. The outward signs of unresponsiveness reflect a deeper transition within neural networks. Consciousness, which depends on coordinated activity across many regions of the brain, begins to dim. This dimming is not a simple switch turning off, but a gradual breakdown of the connections and rhythms that allow perception, thought, and identity to exist together in a unified state. In a healthy brain, consciousness arises from the interaction of widespread neural circuits. Signals move between the cerebral cortex, which supports perception and reasoning, and deeper structures such as the thalamus and brainstem, which help regulate alertness and relay information. This interaction forms a dynamic system in which sensory input is processed, internal states are monitored, and responses are generated. The system requires continuous energy and precise timing. Even small disruptions can affect clarity, attention, and awareness. When fuel loss becomes severe, the system can no longer maintain its integrated activity. The earliest effects often involve confusion and reduced clarity. Thoughts may become disorganized. Attention may narrow or drift. External stimuli, such as sound or touch, may no longer be interpreted in a meaningful way. These changes correspond to reduced coordination between cortical regions. Neurons may still fire, but their activity is less synchronized. Communication between areas weakens. The brain begins to lose its ability to combine information into a coherent experience. As the process continues, awareness fades further. The person may no longer respond to their surroundings in any observable way. This state is commonly described as unconsciousness. Scientifically, it reflects the loss of large-scale network activity that supports wakefulness and perception. The reticular activating system, a group of structures in the brainstem that helps maintain alertness, becomes less effective as oxygen and energy decline. Without its influence, the cortex cannot sustain the patterns required for conscious experience. Electrical activity within the brain changes during this stage. In normal wakefulness, the brain shows complex variable patterns of electrical signals that reflect active processing. As consciousness fades, these patterns often become slower and less organized. In some cases, there may be brief bursts of activity, but they do not form the stable networks needed for awareness. Eventually, measurable electrical activity diminishes significantly. However, the exact relationship between electrical signals and subjective experience remains an area of study. Observations indicate that organized consciousness depends on specific patterns, not just the presence of activity. Memory formation is also affected. The hippocampus, a structure essential for forming new memories, is highly sensitive to oxygen deprivation. As its function declines, the ability to store new information ceases. Even if some awareness persists briefly, it may not be retained. This contributes to the absence of recall in many cases where consciousness is lost rapidly. The breakdown of memory processes reflects the vulnerability of certain neural circuits to energy failure. Sensory perception diminishes alongside awareness. Vision may fade as the visual cortex and associated pathways lose function. Hearing may persist slightly longer in some cases, but its interpretation becomes unclear as higher processing centers fail. Touch, smell, and taste similarly lose meaning as the brain can no longer integrate signals into perception. The body may still receive stimuli, but the brain does not organize them into experience. Motor control also declines. Voluntary movement depends on coordinated signals from the brain to muscles. As neural pathways fail, this control is lost. The body becomes still, not because muscles are incapable of movement, but because the brain no longer directs them in an organized way. Reflexes may persist briefly, as they are mediated by simpler neural circuits. But even these fade as the overall system deteriorates. The sense of self, which depends on continuous integration of memory, perception, and internal awareness, becomes increasingly unstable. Scientific understanding of this process is limited, as subjective experience cannot be directly measured. However, it is known that the brain regions involved in self-awareness, including parts of the cortex, are highly dependent on coordinated activity. As this coordination fails, the processes that support identity and continuity are disrupted. Observations indicate that the conditions required for a stable sense of self are no longer present. Despite these changes, some uncertainty remains. Reports from individuals who have been resuscitated after brief periods of cardiac arrest sometimes include descriptions of experiences during unconsciousness. These accounts vary widely and are not always consistent with measurable brain activity. Research continues to examine how such experiences relate to the final patterns of neural activity. What is clear is that sustained consciousness requires organized, energy-dependent processes that cannot continue without adequate blood flow. Temperature and other factors can influence the rate at which consciousness fades. Lower temperatures reduce metabolic demand and can slow the progression of neural failure. Certain medical conditions or interventions may also alter the timeline. However, under typical conditions, the loss of consciousness occurs rapidly after circulation ceases, often within a short span of time. This reflects the brain's dependence on continuous support. At this stage, the body no longer presents outward signs of awareness. The transition from partial responsiveness to complete unresponsiveness is complete. Internally, the brain's networks have lost the coordination required for conscious experience. The electrical and chemical processes that support perception, thought, and identity are no longer functioning in an organized way. The brain remains as a physical structure, but its role as the center of awareness has effectively ended. This inward region defines the collapse of consciousness as a biological process. It is not described in terms of meaning or interpretation, but in terms of measurable changes in neural activity and metabolism. The dimming of consciousness follows the loss of fuel and the breakdown of network coordination. It marks the point at which the brain can no longer sustain the processes that give rise to awareness, leaving only the remaining physical structures as the process of death continues deeper into the body. After consciousness fades, and the brain can no longer sustain the broad, integrated activity required for awareness, there remains a narrower and more uncertain region. This region concerns the final brain activity that may continue for a brief time after circulation stops. It lies deeper than outward response, deeper than ordinary thought, and deeper than the clear loss of consciousness. Here science looks not at lived experience in a direct way, because lived experience cannot be measured from the outside, but at electrical patterns, chemical shifts, and the last fragments of organized or semi-organized neural function. This is one of the most closely studied and most carefully interpreted parts of the dying process, because it stands at the edge of what can be observed while approaching what remains uncertain. The brain is an electrical organ. Neurons communicate through changes in voltage across their membranes and through the release of neurotransmitters at their connections. In life, this creates a constant field of activity that can be measured at the scalp with electroencephalography, a method that records broad patterns of electrical change. In ordinary wakefulness, these patterns are variable, layered, and responsive. They shift with attention, sleep, sensation, and thought. As the brain loses oxygen and glucose, those patterns become slower and less stable. Eventually, they may become very small or disappear from standard recording. Yet the disappearance of ordinary surface patterns does not always mean every neuron has stopped all activity in the same instant. The brain is not a single lamp going dark. It is a vast and uneven tissue whose regions fail at different rates. Studies in humans and animals show that shortly after circulation ceases, there can still be residual electrical events. Some are brief waves of activity, some are local bursts. Some may reflect the final coordinated discharge of stressed neurons before function collapses further. In some settings, researchers observe a transient rise in certain frequencies of brain activity in the first seconds after cardiac arrest. These signals have drawn attention because they raise the possibility that the dying brain may pass through a short phase of unusual network behavior. What science can say with confidence is limited. These signals are real measurements in some studies, but their meaning is not fully established. They do not prove preserved awareness, and they do not prove the absence of awareness. They show that dying brain tissue can remain electrically active in brief, changing ways after the loss of organized circulation. One important event studied in this context is spreading depolarization. This term describes a large wave of electrical failure that moves through vulnerable brain tissue when cells can no longer maintain their normal ion balance. Under healthy conditions, neurons keep a difference in charge across their membranes. That difference is essential for signaling. When energy stores fail, cells lose the power to maintain it. A broad wave of collapse can then pass through the tissue, during which neurons depolarize and normal signaling breaks down. In some injured but still living brains, such waves can occur and recovery may follow if blood flow is restored. In the dying brain, however, spreading depolarization may become terminal. It marks a point where large regions lose the electrical conditions required for organized function. This is not a dramatic flash in the ordinary sense. It is a measurable physiological event, a final breakdown in the brain's ability to preserve order. Before that terminal breakdown fully unfolds, some neural circuits may continue firing in isolated or unstable ways. The brainstem, deeper nuclei, and fragments of cortical tissue may retain function slightly longer than the larger networks that support consciousness. Activity can therefore persist without forming a coherent mind. This distinction is central. Science does not define consciousness by any electrical activity at all. It depends on sustained, integrated, large-scale coordination across brain systems, a few remaining signals, or even a brief burst across some regions, do not by themselves mean that perception or self-awareness remains intact. They show only that death at the neural level is a process with stages rather than an indivisible instant. This region also intersects with reports from people who are resuscitated after cardiac arrest. Some describe vivid perceptions, unusual clarity, or experiences that they associate with the period near death. These reports are important to researchers because they concern a time when blood flow to the brain is severely reduced or absent. However, the exact timing of such experiences remains difficult to establish. They may occur during the descent into unconsciousness, during brief residual activity after arrest, during early recovery when circulation returns, or through memory processes that later organize fragmentary experience. Observations indicate that near-death reports are genuine reports of experience, but it remains uncertain precisely when and how those experiences arise in relation to measured brain events. Scientific caution is essential here. The existence of reports does not allow a firm conclusion that full consciousness continues after the brain has lost organized function. Chemical events continue shaping this final activity. As oxygen disappears and acidity rises, neurons release neurotransmitters in abnormal amounts. Calcium enters cells excessively. Membranes weaken, supporting cells called glia, which normally help maintain the neural environment, also begin to fail in their stabilizing roles. The tissue becomes less able to separate signal from noise, order from disorder. In this unstable state, brief electrical events may still occur, but they take place in a brain losing the conditions that make meaningful processing possible. The final activity is therefore not simply the last ordinary thought. It is activity within a system whose balance is dissolving. Measurement itself has limits. Electroencephalography mainly captures broad cortical patterns and may miss small, deep, or local events. Clinical monitoring during emergencies is often incomplete, and the timing of cardiac arrest, loss of blood pressure, and recorded brain signals may not align with perfect precision. Different studies use different methods, and the number of human cases with detailed measurement remains limited. For these reasons, scientific conclusions stay modest. There is evidence for brief residual and sometimes complex brain activity near the end of life. There is evidence that terminal waves of depolarization mark a final loss of electrical order. There is not enough evidence to state with certainty that such activity corresponds to sustained conscious awareness. In this way, the final brain activity stands at a boundary between the measurable and the unresolved. It is measurable because electrodes can detect patterns, because tissue physiology follows known principles, and because the dying brain has been studied in laboratory and clinical settings. It is unresolved because subjective experience cannot be read directly from those measurements, especially during a period when ordinary neural organization is rapidly failing. Science can describe the patterns and their likely physical meaning, but it cannot fully translate them into inner experience. So this inward layer remains narrow and quiet. After the larger architecture of consciousness has dimmed, the brain may still produce brief signals, local bursts, or terminal waves as its cells lose the ability to hold their electrical balance. These are the last measurable traces of neural function, not a restored mind, and not proof of what the person experiences. Observations indicate that the brain does not cease all activity in a single instant. It remains uncertain whether the final fragments of activity support any awareness at all. The scientific answer in this region is therefore precise in physiology and restrained in interpretation, holding to what can be observed while leaving unresolved what cannot yet be known. Beyond the final measurable bursts of brain activity, the scientific view moves inward again, from organs and networks to the level of individual cells. At this depth, death is no longer mainly a matter of fading awareness or failing circulation. It becomes a crisis of structure and balance inside living tissue. A cell is not alive simply because it exists as a small container of matter. It remains alive because it actively preserves an unstable order. It keeps certain chemicals concentrated inside and others outside. It maintains voltage across its membrane. It controls water, acidity, temperature, repair, and waste removal. All of this requires energy. When the supply of oxygen and nutrients has been cut off, and the larger systems of the body have stopped supporting life, cells begin to enter a state in which that order can no longer be maintained. In life, each cell is enclosed by a membrane, a thin boundary that separates its interior from the surrounding fluid. This membrane is not a passive wall. It contains channels, pumps, and receptors that regulate what enters, what leaves, and how the cell communicates. One of its most important tasks is to maintain differences in sodium, potassium, calcium, and other charged particles between the inside and outside. These differences are essential for nerve signaling, muscle contraction, transport of nutrients, and many other functions. They are held in place by proteins that consume energy continuously. When death interrupts circulation, oxygen delivery stops. Glucose is no longer replenished, and the cell's ability to make energy falls. As a result, the membrane begins to lose control. This failure does not happen at the same speed in every tissue. Some cells are more resistant than others. Skin cells, some connective tissue cells, and parts of bone can remain structurally intact for long periods even after the organism has died. Brain cells, by contrast, are highly sensitive and begin to fail quickly. Heart muscle cells are also vulnerable. Still, the pattern is shared across the body. Energy production declines, ion balance weakens, and the membrane becomes less able to protect the cell's internal environment. This is the beginning of cellular crisis. One of the earliest changes is the failure of ion pumps. These molecular machines normally move sodium out of the cell and potassium in, preserving the gradient needed for electrical stability and fluid balance. Without adequate energy, the pumps slow and then stop. Sodium begins to accumulate inside the cell. Potassium leaks outward. Water follows sodium, because water moves toward areas with higher concentrations of dissolved particles. The cell starts to swell under a microscope. This swelling is one of the classic early signs of injury. It reflects a simple fact. The cell can no longer regulate its internal chemistry against the pull of physics. Calcium also becomes a major part of the crisis. In healthy cells, calcium levels are kept very low inside the main body of the cell and stored carefully in compartments or kept outside the membrane. Calcium is used as a signal, a brief messenger that can trigger contraction, secretion, or enzyme activity. During cellular failure, calcium floods into places where it should not remain. This excess calcium activates enzymes that begin to damage proteins, membranes, and internal structures. What was once a useful signal becomes part of a destructive process. The cell's own machinery, designed for controlled living function, begins to operate in uncontrolled and harmful ways. Mitochondria, the structures that produce much of the cell's usable energy, are central in this stage. They rely on oxygen to support efficient energy production. Without oxygen, they cannot maintain normal function. Their membranes become damaged, their output falls, and they may begin releasing signals associated with cell death pathways. Mitochondria are not merely power sources, they also help regulate survival and stress responses. As they fail, the cell loses both energy and the ability to control damage. The internal environment becomes increasingly disordered. Acidity rises as well when oxygen is absent. Some cells shift briefly to a less efficient form of metabolism that produces lactic acid. At the same time, waste products are no longer carried away by blood. The result is a more acidic environment inside tissues. Many enzymes function only within a narrow chemical range. As acidity changes, proteins lose their usual shape and efficiency. Reactions that support life slow down or stop. Other reactions, including destructive ones, may increase. A living cell depends on fine chemical balance. Death removes the conditions that keep that balance in place. The internal skeleton of the cell also begins to weaken. Cells are supported by a framework of protein fibers that help maintain shape, position, and transport within the cell. This structure is called the cytoskeleton. It allows organelles to move, membranes to hold their form, and tissues to resist stress. As energy fails and destructive enzymes become active, the cytoskeleton breaks down. The cell loses its shape and its internal organization. Surface projections may flatten. Junctions between cells weaken. In tissues that depend on precise architecture, such as the lining of the gut or the filtering structures of the kidney, this loss of order has rapid consequences. Communication between cells also deteriorates. Living tissue depends on constant signaling. Hormones, neurotransmitters, local chemical messengers, and electrical impulses all help cells act as part of a larger whole. After death, those signals become fragmented. Some receptors still exist for a time, and some cells may still respond weakly to local conditions, but the integrated conversation of tissue is over. A heart muscle cell without circulation is no longer part of a beating organ. A neuron without fuel is no longer part of a working network. At the cellular level, death is the loss of cooperative order as much as the loss of individual stability. Not all cells die through exactly the same mechanism. Some follow a pathway called necrosis, in which injury becomes so severe that the membrane breaks, contents leak out, and surrounding tissue is damaged. Others may briefly enter programmed pathways associated with controlled cell death, especially if the injury develops less suddenly. But after the death of the whole organism, uncontrolled injury becomes dominant. There is no restored circulation, no immune regulation, no repair system strong enough to contain the damage. Cellular survival becomes temporary and local, not part of a living body. This stage reveals an important scientific truth. Death is not one event repeated identically in every part of the body. It is a layered breakdown in which cells respond according to their needs, their structure, and the conditions around them. Some tissues remain viable for transplantation for a limited time. Some sperm, skin, or bone cells can persist longer than the person as a whole has remained alive. Yet this does not reverse the larger fact. The organism has already died because coordination among organs has ended. What remains is the gradual failure of the parts. So in this inward region, death becomes microscopic. The surface still resembles a body, and even organs still retain their form. But within each tissue, the struggle to maintain order is failing. Membranes lose selectivity. Ion gradients collapse. Water shifts inward. Calcium triggers damage. Mitochondria fall silent. Acidity rises. The framework of the cell weakens. Each living unit moves from controlled function into chemical instability. This is the cellular crisis of death. The stage in which life is no longer lost only at the scale of breath, blood, or brain, but within the fundamental compartments from which the body is built. As cellular stability declines, the next inward region is defined by chemistry that no longer serves life. The body remains composed of the same molecules it held during life, yet the way those molecules interact begins to change. In a living state, chemical reactions are tightly controlled. Enzymes guide them, membranes compartmentalize them, and energy directs them toward repair, growth, and balance. After death, this control fades. The chemistry does not stop, but it becomes unregulated. Reactions continue without coordination, and the internal environment shifts steadily away from the conditions required for living function. One of the earliest and most consistent changes is the progression of acidity. During life, the body maintains a narrow range of chemical balance, especially in the blood and within cells. This balance is essential for proteins to keep their shape and for enzymes to work efficiently. When circulation fails and oxygen is no longer supplied, cells rely briefly on less efficient metabolic pathways that produce acidic byproducts. At the same time, waste products that would normally be removed begin to accumulate. Without blood flow, there is no system to carry these substances away or to buffer their effects. The result is a gradual increase in acidity throughout tissues. This shift in acidity has wide consequences. Proteins depend on their three-dimensional structure to function. Changes in acidity alter the electrical charges on these molecules, causing them to fold differently or lose their shape. When this happens, enzymes can no longer guide reactions in a controlled way. Structural proteins that support cells and tissues weaken. Membrane proteins that regulate transport lose precision. The entire chemical system begins to drift away from the ordered state that defines life. Enzymes themselves become part of the process. In a living body, enzymes operate within defined compartments and under strict regulation. Many are contained within structures such as lysosomes, which are small organelles that store substances capable of breaking down cellular material. These enzymes are essential for recycling damaged components and maintaining cellular health. After death, as membranes lose integrity, these enzymes are released into areas where they are not normally active. They begin to act on proteins, lipids, and other molecules without the usual controls. This marks the beginning of widespread internal breakdown driven by the body's own chemistry. Energy molecules such as adenosine triphosphate are no longer produced in sufficient quantities. Without energy, many chemical reactions that require active control cannot proceed. Processes such as maintaining ion gradients, repairing damage, and synthesizing new molecules come to a halt. What remains are reactions that can occur without energy input, or that are driven by the new chemical conditions. These include breakdown reactions that dismantle complex structures into simpler components. The direction of chemistry shifts from construction and maintenance toward disassembly. Lipids, which form the basis of cell membranes, begin to degrade. Membranes are made of lipid layers interspersed with proteins, and their stability depends on both structure and environment. As acidity rises and enzymes become active in uncontrolled ways, lipids can be broken down into smaller molecules. This weakens the barriers that separate different parts of the cell and different tissues. As membranes deteriorate, the contents of cells mix more freely with their surroundings. The distinction between compartments fades, and chemical gradients that once defined a living organization disappear. Proteins undergo similar changes. They are broken into smaller fragments by enzymes that are no longer confined. Structural proteins that give tissues their strength begin to lose integrity. Enzymatic proteins that once guided reactions are themselves degraded. The breakdown of proteins contributes to the overall loss of structure within tissues. It also releases amino acids and other small molecules into the surrounding environment, further altering the chemical composition of the body. Nucleic acids, including DNA and RNA, are also affected. These molecules carry genetic information and guide the production of proteins. After death, they're gradually broken down by enzymes. While DNA can persist for some time under certain conditions, especially in protected environments, its integrity declines as chemical reactions proceed. The information it carries is no longer used by living systems, and the processes that once read and replicate it have stopped. Water distribution changes as well. In living tissue, water is carefully regulated, with different concentrations inside and outside cells. As membranes lose control and proteins degrade, water moves more freely. Cells that have swollen due to ion imbalance may eventually rupture, releasing their contents. The surrounding tissue becomes more uniform in composition, losing the distinct boundaries that once separated functional units. The overall effect of these changes is a transition from organized chemistry to passive chemistry. In life, Reactions are directed toward maintaining a stable internal state, known as homeostasis. After death, there is no longer a system to enforce this stability. Reactions proceed according to basic physical and chemical principles without guidance toward a living goal. The body becomes a collection of substances undergoing change rather than a coordinated system maintaining itself. Temperature influences the rate of these chemical processes. Higher temperatures generally accelerate reactions, leading to faster breakdown. Lower temperatures slow them, preserving structure for a longer period. This is why cooling can delay some aspects of postmortem change, and why environmental conditions play a significant role in how quickly the body's chemistry evolves after death. However, regardless of temperature, the direction remains the same. Controlled chemistry gives way to uncontrolled change. Oxygen, though no longer delivered through circulation, may still be present in small amounts within tissues or the surrounding environment. In some cases, this can lead to oxidative reactions, where molecules react with oxygen and become altered. These reactions can contribute to the degradation of lipids and proteins. Without the protective systems that exist in living cells, such as antioxidants and repair enzymes, these reactions proceed unchecked. At this stage, the body is still recognizable as a whole, but its internal chemistry is no longer aligned with life. The processes that once sustained function have been replaced by processes that dismantle structure. The change is gradual and continuous, not marked by a single moment. Each reaction contributes to a broader shift in the state of the tissue, moving it further from the organized condition of life. Scientific understanding of this region is well established. The chemical pathways involved are known, and their progression has been observed in many contexts. What remains variable is the exact timing, which depends on factors such as temperature, cause of death, and environmental conditions. Observations indicate that these chemical changes begin soon after death and continue as long as conditions allow. Thus, the chemistry of early death is defined by loss of control. Acidity rises, enzymes act without regulation, membranes weaken, and molecules break down. The body's internal environment shifts from a balanced system to one governed by passive reactions. This marks a deeper stage in the process of dying, where life is no longer sustained even at the level of chemical organization, and the transformation of the body continues inward and onward. After the early chemical changes have begun inside tissues, the next region is not deeper within a particular organ, but broader again in physical process. The body begins to lose heat. In life, warmth is not a static condition. It is continuously produced and continuously regulated. Cells generate heat as they use energy. Muscles contribute through movement and tone. The liver and other organs, active in metabolism, release warmth into the blood. Circulation distributes that warmth through the body, and the nervous system helps maintain a narrow internal temperature by adjusting blood flow, sweating, shivering, and behavior. After death, this internal production and control cease. The body remains warm for a time, but the warmth is no longer renewed. It begins to drift toward the temperature of the surrounding environment. This process is known as algor mortis, a Latin term used in forensic science to describe the cooling of the body after death. The term is useful, but the process itself is simple. Heat moves from warmer regions to cooler ones. A living human body usually maintains an internal temperature higher than the surrounding air in most ordinary conditions. Once metabolism stops and blood no longer circulates, the body can no longer resist that temperature difference. Heat leaves by conduction, which is direct transfer to surfaces in contact with the body, by convection, which is transfer to moving air or water, by radiation, which is emission of heat energy into the surroundings, and sometimes by evaporation if moisture is present. These mechanisms are present during life as well, but in life they are balanced by ongoing heat production. After death, only loss remains. The first important point in this region is that cooling does not happen all at once. The body is large, layered, and uneven. The skin begins to approach the temperature of the environment sooner than deeper tissues. Hands, feet, ears, and face often cool early because they are exposed and because circulation has already been reduced there during the final stages of dying. The inner organs and central tissues stay warm longer. The body becomes thermally uneven, with outer regions cooling while the core retains heat for a time. In life, blood circulation helps reduce such differences by carrying warmth from the core outward. After death, that distribution stops. Temperature becomes a passive matter of tissue depth, insulation, and exposure. Clothing, bedding, body size, air movement, humidity, and contact with surfaces all influence the rate of cooling. A body in still, warm air loses heat more slowly than a body in cold moving air. A body lying on a metal surface cools faster than one resting on thick fabric or dry soil, because some materials conduct heat more efficiently than others. Water removes heat especially quickly. This is why immersion in cold water can lower body temperature much faster than exposure to cold air. Fat beneath the skin can act as insulation, slowing heat loss somewhat. Thin bodies, unclothed bodies, and bodies in windy or wet conditions cool more rapidly. These are not secondary details. They are part of the basic physics that shapes what happens after death. Because cooling follows physical laws, it has long been used in attempts to estimate the time since death. Forensic investigators may measure body temperature and compare it with the surrounding environment. Under controlled conditions, this can provide useful information. But scientific practice is careful here, because the process is influenced by many variables. There is no universal rate that applies to every case. A body may begin cooling quickly or slowly depending on the cause of death. The setting, the amount of insulation, prior body temperature, illness, fever, or environmental exposure. Observations indicate that cooling follows a general trend, but it remains uncertain exactly how fast it will proceed in any individual case without considering the full context. The loss of heat also marks an important biological boundary. A living body does not merely contain warmth, it generates and defends warmth. Temperature regulation is an active achievement, maintained by the brain, endocrine system, blood vessels, sweat glands, and muscles. After death, this achievement ends. The body no longer shivers to make heat, no longer narrows vessels to conserve it, and no longer adjusts its position or behavior in response to cold. Heat leaves according to the environment alone. In this sense, cooling is not just a physical consequence of death. It is evidence that one of the body's most basic regulatory powers has ceased. At the tissue level, cooling changes the speed of other processes already underway. Chemical reactions generally slow as temperature falls. Enzyme activity becomes less efficient. The breakdown of membranes and proteins continues, but more slowly in colder conditions. This is why cooling can delay decomposition and preserve tissue structure for a time. Refrigeration after death does not stop change completely, but it reduces its pace. In warm environments, by contrast. Early post-mortem chemistry proceeds faster. Thus heat loss is both an effect of death and a factor that shapes what follows. The experience of touch reflects this transition in a direct way. During life, skin feels warm because blood flow and metabolism maintain a moving source of heat beneath the surface. After death, even before the body fully cools, the quality of that warmth changes. It is no longer being renewed. A warm body shortly after death is already thermally passive. This difference may not be visible, but it is real. The heat present in the tissues is now a stored remainder, not a product of ongoing life. Over time, that remainder drains into the surroundings. Temperature gradients inside the body also change in predictable ways. The head and trunk, which contain the brain and major organs, often retain heat longer than the limbs. Areas in contact with cold surfaces may cool asymmetrically. If the body is turned or moved, the pattern of heat loss may shift. In some cases, pockets of warmth may persist internally even after the skin feels cool. This unevenness is one reason simple external touch cannot precisely determine how long a body has been dead. Scientific measurement requires thermometers, context, and careful interpretation. The environment does not act on the body with intention. It simply receives the heat the body no longer keeps. Air carries it away. Surfaces absorb it. Water strips it rapidly from exposed tissue. The body begins to settle into the temperature of the world around it, just as it will later settle into its chemical and biological cycles. This stage, therefore, has a quiet clarity. It is the moment when the body stops being a source of warmth and becomes an object subject to ordinary heat exchange. There are unusual situations that complicate this picture. Severe fever before death can leave the body starting from a higher temperature. Hypothermia before death can mean the body is already cold at the moment of death. Some diseases, drugs, or environmental conditions alter circulation and muscle activity in ways that affect how much heat is present at the start of the post-mortem period. Yet these variations do not change the underlying rule. Once metabolism and regulation cease, body temperature moves toward ambient temperature, the temperature of the surrounding environment. So in this region, death is expressed through the fading of internal warmth into external balance. The body no longer makes heat, no longer distributes it with blood, and no longer protects it through regulation. The outer layers cool first, the deeper tissues later, and the whole body gradually approaches the temperature of air, water, fabric, soil, or whatever surrounds it. Heat leaves by ordinary physical pathways, not by any hidden force. The living warmth of the body becomes residual warmth, and residual warmth becomes environmental equilibrium. This is the cooling of death, a steady return from active internal temperature to the quiet temperature of the world. As the body continues to cool and its internal chemistry shifts further from the conditions of life, physical changes in muscle and blood distribution begin to appear more clearly. These changes are not signs of new activity, but consequences of the loss of circulation, energy, and control. Two of the most well-known processes in this region are rigor mortis and livor mortis. Together, they describe how the body's form becomes fixed and how blood settles under the influence of gravity after the heart has stopped. Rigor mortis refers to the stiffening of muscles after death. In life, muscle fibers contract and relax through a carefully regulated cycle that depends on energy. Within each muscle cell, proteins called actin and myosin interact to produce contraction. This interaction requires energy in the form of adenosine triphosphate. When energy is available, muscles can both contract and release, allowing movement and flexibility. After death, energy production ceases. As the remaining energy stores are used up, the chemical conditions inside muscle cells change. Calcium levels rise within the cells, triggering contraction, but without energy, the proteins cannot separate again. The result is a sustained state of contraction that leads to stiffness. This stiffness does not appear immediately at the moment of death. It develops over time as the chemical processes within muscle cells progress. Typically, muscles begin to stiffen within a few hours, though the exact timing depends on temperature, physical condition, and other factors. The process often starts in smaller muscles, such as those of the face and jaw, and then spreads to larger muscle groups in the limbs and trunk. This sequence reflects differences in muscle size, structure, and metabolic activity. Over time, the entire body may become rigid. Rigor mortis is temporary. As decomposition advances and proteins begin to break down, the stiffness gradually disappears. Enzymes and microbial activity weaken the structures that maintain the contracted state. Muscles lose their integrity, and the body becomes flexible again. This progression from flexibility to stiffness and back to flexibility is a well-documented pattern in postmortem change. It provides useful information in forensic contexts, but it also illustrates the underlying biology of muscle function and its dependence on energy. At the same time, blood within the body undergoes its own transformation. Without the pumping action of the heart, blood is no longer circulated through vessels. Instead, it begins to settle under the influence of gravity. This process is called lever mortis. Blood collects in the lower parts of the body, depending on the position in which it lies. Areas in contact with the ground or pressed against surfaces may appear pale because pressure prevents blood from pooling there, while exposed lower regions develop a darker coloration. The color changes associated with liver mortis are due to the accumulation of a deoxygenated blood in small vessels and capillaries. Without circulation, oxygen is no longer replenished, and the blood takes on a darker tone. The skin above these areas reflects this change, often appearing purplish or bluish. This is not an active process, but a passive settling of fluid within the vascular system. Over time, the pattern becomes more defined as blood continues to move downward and remains in place. Like rigor mortis, liver mortis develops gradually. It may begin within a short time after death and become more pronounced over several hours. Initially, the discoloration can shift if the body is moved, because the blood has not yet fully settled. Later, as the blood coagulates and tissues begin to fix in place, the pattern becomes stable. This stabilization reflects ongoing chemical changes within the blood and surrounding tissues. The interaction between rigor mortis and liver mortis shapes the body's appearance during this stage. Stiff muscles hold the body in a fixed posture, while gravity determines where blood accumulates. Together, these processes create a new physical state that is distinct from both living movement and the earlier stillness of death. The body becomes an object governed by physical forces rather than biological control. Temperature continues to influence these changes. Warmer conditions accelerate the chemical reactions that lead to rigor mortis and the settling of blood. Cooler conditions slow them. This variability is important in understanding how different environments affect the timeline of postmortem processes. Despite these differences, the underlying mechanisms remain consistent. They are driven by the loss of energy, the effects of gravity, and the ongoing chemical changes within tissues. Other physical effects may accompany these processes. Fluids can shift within the body as pressure changes and tissues lose their structural integrity. Small amounts of fluid may leak from vessels into surrounding areas. The skin may begin to lose elasticity as underlying structures weaken. These changes are part of the broader transition from a living system to a passive physical state. At this stage, the body's interaction with its environment is defined by physical laws rather than biological regulation. Muscles no longer respond to signals from the brain. Blood no longer moves through vessels in a controlled circuit. Instead, The body's form and appearance are shaped by gravity, chemistry, and the gradual breakdown of cellular structures. The processes of rigor mortis and livor mortis are clear expressions of this shift. Scientific understanding of these processes is well established. They have been observed across many conditions and studied in detail. While the exact timing can vary, the sequence and mechanisms are consistent. Rigor mortis reflects the loss of energy and the resulting fixation of muscle proteins. Livor mortis reflects the passive movement of blood under gravity. Both are predictable consequences of the end of circulation and metabolism. In this inward region, death is expressed through the body's physical settling. Muscles become fixed as energy disappears. Blood pools where gravity directs it. The body's posture and coloration change in ways that reflect the absence of life's active control. These processes do not restore or alter the fact of death. They reveal it more clearly, showing how the body transitions from a coordinated organism into a structure shaped by the basic forces of physics and chemistry. After the body has cooled, after muscles have stiffened and blood has settled, the next inward region is defined by the body's own internal breakdown. This process is called autolysis. The term means self-digestion. It does not describe a living act of repair or recycling, but the release and spread of enzymes that begin dissolving cells and tissues from within once the systems that normally contain them have failed. In life, cells use enzymes to build, dismantle, and renew their components in tightly controlled ways. These enzymes are essential to health. After death, as membranes weaken and compartments break apart, the same chemical tools begin acting without regulation. The body starts to digest itself. Autolysis begins soon after death. Though it's not always visible from the outside in its earlier stages, it starts at the cellular level, where membrane failure and chemical imbalance have already prepared the way. In living cells, many destructive enzymes are stored inside lysosomes, small membrane-bound structures that contain substances capable of breaking down proteins, lipids, carbohydrates, and nucleic acids. This storage is safe while the membrane remains intact and while the cell continues to regulate acidity and transport. After death, those protections fail. Lysosomal membranes become more fragile. The acidity within cells changes. Enzymes escape into the surrounding interior and begin digesting cellular material. This self-digestion affects different tissues at different speeds. Organs rich in enzymes and water tend to break down earlier and more quickly than drier or more fibrous tissues. The pancreas is one of the clearest examples, because it contains strong digestive enzymes even in life. These enzymes normally help break down food in the intestine, but after death they can contribute to rapid tissue destruction within the organ itself and nearby structures. The stomach and intestines also begin to lose their internal lining as enzymes and acid are no longer contained by living cells. The liver, dense with metabolic activity in life, undergoes marked autolytic change as its cells lose structure. By contrast, tissues such as tendons, ligaments, and some parts of the uterus or prostate may remain structurally intact for longer periods because they contain less water and fewer active enzymes. The mechanism is steady and local. Proteins that form the framework of the cell begin to break apart. Membranes lose integrity and allow fluids and molecules to move more freely. Internal compartments, which in life separate one reaction from another, collapse. The nucleus, which contains genetic material, begins to change. Its membrane weakens, and enzymes degrade DNA and associated proteins. Under the microscope, pathologists observe a progression. Nuclei may shrink, then fragment, then disappear. The cell's interior becomes more uniform, less organized, and more pale or granular in appearance as its structures dissolve. Water plays an important role in autolysis, because many tissues contain large amounts of water. Enzymatic reactions can continue in the moist internal environment, even after the organism is dead. Cells that have already swollen from ion imbalance may rupture, releasing more enzymes into the surrounding tissue. This adds to the breakdown. The process is therefore not confined to isolated cells. Damage in one area can spread chemically into nearby areas as membranes and boundaries continue to fail. The body's microscopic organization begins to blur. Acidity continues to shape the pace and pattern of autolysis. Enzymes function differently at different levels of acidity, and the post-mortem environment often favors those that dismantle tissue. Since blood is no longer circulating, there is no mechanism to buffer local chemical conditions. Waste products accumulate. Acidic environments develop in many regions. These conditions promote the activity of some lysosomal enzymes and contribute to the continued dissolution of cellular components. In this sense, autolysis grows directly out of the earlier chemistry of death. The rising acidity, failing membranes, and loss of energy all feed the same process. Certain organs show this transformation in ways that later become visible. The lining of the stomach may soften and detach. The inner surface of blood vessels may lose its cellular layer. The cornea of the eye can become cloudy because its cells are no longer maintained and fluid balance has failed. Mucosal surfaces, which in life renew themselves quickly and depend on active transport, deteriorate relatively early. Yet even where the changes remain hidden, the principle is the same. Tissue is no longer preserved by life. It is being reduced by its own chemistry. Autolysis does not require bacteria to begin. This is an important distinction. It is driven first by the enzymes already present inside the body's own cells. Microorganisms will become major agents later, but in this region the breakdown can be explained without them. The body contains within itself the means of its own early dissolution. This is not unusual in biology. Living systems always contain molecules capable of destroying their parts, because controlled breakdown is necessary for renewal, defense, and digestion. What changes after death is not the existence of these molecules, but the loss of boundaries and regulation that once kept them serving life. Temperature affects autolysis strongly. Warm conditions speed enzymatic reactions and membrane failure, leading to faster tissue breakdown. Cold conditions slow the process, sometimes significantly. Refrigeration after death is therefore used in hospitals and morgues to delay visible decomposition, largely by slowing autolysis and later microbial growth. Still, even in cold conditions, autolysis is delayed rather than fully prevented. As long as tissue remains moist and enzymes remain chemically active, some degree of self-digestion continues. The study of autolysis also matters in medicine and forensic science, because it can resemble disease under the microscope if not interpreted carefully. Some tissue changes seen after death can look similar to injury that occurred during life. Pathologists must distinguish between true disease processes and post-mortem self-digestion. This is done by considering timing, location, surrounding tissue response, and the absence of signs of inflammation, which is the living body's reaction to injury. After death, there is no active inflammatory response. Tissues can break down, but they cannot mount repair or defense. This helps separate autolysis from processes that happened before death. At the larger scale, autolysis softens the distinction between organ and environment within the body. In life, each organ has defined architecture, selective barriers, and precisely maintained fluids. After death, those distinctions begin to fade. The stomach no longer contains acid safely behind living mucosa. The pancreas no longer stores digestive enzymes within stable acinar cells, which are the cells that make those enzymes. The liver no longer preserves the ordered plates of cells that guide its many chemical tasks. The body is still recognizably human in shape, but internally it is becoming less compartmentalized and less controlled. This inward stage is quiet but decisive. No heartbeat is required for it. No brain activity guides it. No outside force initiates it. The body begins to digest itself because the mechanisms of separation and control have ended. Enzymes leak, membranes dissolve, nuclei fragment, tissues soften. What had been an organized collection of living cells becomes a chemically active mass in which structure is steadily lost from within. Autolysis is the body's own first sustained work of decomposition, the process by which death moves deeper into matter and begins turning living tissues into simpler forms. As autolysis progresses and the body's own enzymes continue to dissolve internal structures, another process begins to take greater influence. Microorganisms, many of which were already present during life, now act without restraint. This stage is defined by decomposition driven by bacteria and other microscopic life forms. These organisms do not arrive as something entirely foreign. Many live within the human body during life, especially in the digestive system, where they play roles in digestion and health. After death, the balance that once kept them contained and regulated disappears. They begin to spread, multiply, and break down tissue as part of their natural metabolism. In a living body, the immune system controls microbial populations. Barriers such as skin and mucous membranes limit their movement. Blood circulation supports immune cells that respond to invasion. After death, these systems no longer function. The immune response stops. Barriers weaken as tissues undergo autolysis. The internal environment changes in ways that favor microbial growth. Nutrients become more available as cells break apart. Oxygen levels may vary, creating conditions for different types of bacteria to thrive. The body becomes a suitable environment for organisms that feed on organic material. The digestive tract is one of the primary starting points for this process. During life, it contains a large and diverse community of bacteria. These microorganisms are normally confined to the intestines, where they assist in breaking down food and producing certain compounds. After death, as the intestinal walls lose integrity through autolysis, bacteria begin to move into surrounding tissues. This movement is not directed or purposeful. It follows the breakdown of physical barriers and the availability of nutrients. From the intestines, bacteria spread into the abdominal cavity and beyond. As bacteria metabolize tissue, they produce gases and chemical byproducts. These include substances such as methane, hydrogen sulfide, and carbon dioxide. The accumulation of gases within the body can lead to swelling, particularly in the abdomen. This swelling is a common feature of decomposition. It reflects the activity of microorganisms breaking down proteins, fats, and carbohydrates. The gases are not simply byproducts, they also influence the environment within the body, affecting pressure and further altering tissue structure. The breakdown of proteins by bacteria produces compounds that contribute to the characteristic odor of decomposition. These include molecules formed from amino acids, such as putrescine and cadaverine. These substances arise from the microbial digestion of tissue and are part of the natural chemical transformation of organic matter. While the presence of odor is a noticeable aspect of this stage, it is not the focus of the process. It is one of many indicators that chemical and biological breakdown is underway. Different types of bacteria contribute to decomposition in different ways. Some thrive in environments with little or no oxygen, known as anaerobic conditions. Others require oxygen and are more active in exposed tissues. The balance between these groups depends on the environment inside and around the body. In buried or enclosed conditions, anaerobic bacteria may dominate. In open environments, aerobic bacteria and other organisms may play a larger role. This variation influences the rate and pattern of decomposition. Insects can also become part of this stage, particularly in exposed environments. Certain species are attracted to decomposing tissue and lay eggs, which develop into larvae. These organisms contribute to the breakdown of soft tissues. Their activity is part of a broader ecological process in which organic matter is recycled. However, the presence and influence of insects depend on environmental conditions and access to the body. In some settings, microbial activity remains the primary driver of decomposition. As microorganisms continue to act, the structure of tissues changes further. Organs lose their form as cells are broken down and replaced by simpler substances. Fluids may accumulate or be released as tissues liquefy. The distinction between different types of tissue becomes less clear. Muscle, fat, and connective tissue all undergo transformation, though at different rates. The body's composition shifts from organized biological structures to a mixture of simpler chemical compounds. Temperature remains an important factor. Warmer conditions generally increase the rate of microbial growth and activity, leading to faster decomposition. Cooler conditions slow these processes. Moisture also plays a role, as many microorganisms require water to function effectively. Dry environments can limit microbial activity and preserve tissue for longer periods. These environmental influences do not change the underlying biology, but affect how quickly it unfolds. The spread of microorganisms throughout the body follows the breakdown of internal barriers. Blood vessels, which once carried immune cells and nutrients, now provide pathways for microbial movement as their walls weaken. Connective tissues, which held organs in place, lose their integrity, allowing further spread. The process is not uniform. Some areas may decompose rapidly, while others remain relatively intact for longer. This uneven progression reflects differences in tissue composition, microbial access, and environmental exposure. At this stage, the body is no longer functioning as an organism in any sense. Its systems have ceased to operate in a coordinated way. Instead, it becomes part of a broader biological cycle. Microorganisms that were once contained now act freely, breaking down complex molecules into simpler forms. This transformation is part of the natural recycling of organic matter in the environment. The elements that made up the body begin to return to the surrounding ecosystem. Scientific understanding of microbial decomposition is extensive. It is studied in fields such as microbiology, ecology, and forensic science. Observations indicate that the sequence of microbial activity follows general patterns, though the details vary with conditions. The process is not random. It is governed by the biological characteristics of microorganisms and the physical environment in which they operate. Uncertainty remains in predicting exact timelines, as many variables influence the rate of decomposition. Temperature, humidity, access to oxygen, presence of insects, and the condition of the body all play roles. Despite this variability, the fundamental process is consistent. Microorganisms break down tissue as part of their metabolism, and in doing so, they transform the body into simpler chemical components. In this inward region, death is expressed through the activity of life at a smaller scale. The body, once maintained by its own systems, becomes a resource for other organisms. bacteria and other microorganisms continue the process begun by autolysis, accelerating the breakdown of tissues and redistributing their components. The transformation is steady and inevitable under natural conditions. It marks the transition from individual biological existence to participation in a wider ecological system. As microbial activity continues and the body structures are reduced to simpler substances, the next region extends beyond the body itself into the surrounding environment. The transformation that began within cells and tissues now becomes part of larger natural systems. The materials that once formed a living human organism begin to disperse into soil, air, water, and other elements of the earth. This stage is not defined by a single pathway, because the physical outcome after death varies widely depending on conditions such as burial, exposure, fire, or immersion. Yet across these different settings, the underlying principle remains consistent. The body's components return to broader environmental cycles. In a natural setting where the body is exposed to soil, decomposition continues through the combined action of microorganisms, insects, and environmental factors. As tissues break down, fluids rich in organic compounds may seep into the ground. These fluids contain nitrogen, phosphorus, carbon, and other elements that were part of the body's structure. Soil microorganisms use these compounds as nutrients, incorporating them into their own metabolism. Plants may also absorb these elements through their roots. In this way, the material of the body becomes part of the nutrient cycles that sustain life in the environment. Carbon, one of the most abundant elements in the human body, follows a clear path in this process. During decomposition, carbon-containing compounds are broken down into simpler molecules, including carbon dioxide. This gas can be released into the air through microbial respiration. In some conditions, methane may also be produced by anaerobic bacteria. These gases enter the atmosphere and become part of the global carbon cycle, where they can be used again by plants during photosynthesis. The carbon that once contributed to a living body thus continues its existence in new forms within the environment. Nitrogen follows a similar path. Proteins and nucleic acids in the body contain nitrogen, which is released during decomposition. Microorganisms convert these compounds into forms such as ammonia, which can then be further transformed into nitrates by soil bacteria. These nitrates are essential nutrients for plants, supporting growth and development. Through this process, nitrogen cycles from the body into the soil and back into living systems. The transformation is part of a continuous exchange that occurs across all ecosystems. Water, which makes up a large portion of the human body, also returns to the environment. As tissues break down, water is released and may evaporate into the air or be absorbed into the ground. In warm conditions, evaporation can occur relatively quickly. In cooler or more humid environments, water may remain within the soil or contribute to local moisture levels. This redistribution of water is part of the broader hydrological cycle, in which water moves between the Earth's surface, atmosphere, and living organisms. Different methods of handling the body after death influence how these processes unfold. In burial, the body is placed within soil, where decomposition proceeds under varying levels of oxygen and microbial activity. The presence of a coffin or other container can slow or alter the process by limiting access to air and organisms. In cremation, high temperatures reduce the body to ashen gases, releasing carbon dioxide and water vapor into the atmosphere. The remaining mineral components, such as calcium and phosphorus, may be retained in ash and later return to the environment. In aquatic environments, decomposition occurs in water, with different microbial communities and rates of change. Each pathway reflects the same underlying transition from organized biological structure to dispersed chemical components. Over longer periods, the more durable elements of the body persist. Bones, composed largely of mineralized tissue, can remain for years or centuries under certain conditions. They gradually break down through physical weathering, chemical dissolution, and microbial action. Calcium and phosphorus from bone eventually enter soil and water systems, contributing to the availability of these elements for other organisms. Even these more resistant structures are not permanent. Given enough time and the right conditions, they too are reduced and redistributed. The environment does not preserve the identity of the body. It transforms its components according to physical and chemical laws. There is no central process directing this transformation toward a specific outcome. Instead, it is governed by interactions between molecules, organisms, and environmental conditions. The body becomes part of the same cycles that affect all organic matter. These cycles are continuous and do not depend on the origin of the material, whether from plants, animals, or microorganisms. Organic matter is broken down and reused. Scientific observation confirms that this return to the environment is universal under natural conditions. The specific details vary, but the overall pattern is consistent. Organic material does not remain isolated indefinitely. It is incorporated into larger systems through decomposition and recycling. This process is essential for maintaining the balance of ecosystems, allowing nutrients to be reused, and supporting the growth of new life. There are conditions that can slow or alter this return. Extremely dry environments can preserve tissue through desiccation, reducing microbial activity. Very cold environments can slow chemical reactions and decomposition. In rare cases, bodies may be preserved for long periods under such conditions. However, these are exceptions rather than the rule. Even in these cases, given enough time, environmental forces will continue to act on the material. In this outward extension of the process, the boundary between the body and the environment becomes indistinct. The materials that once formed a single organism are no longer contained within it. They are distributed through air, soil, and water, entering new contexts and participating in ongoing cycles. The body is no longer a separate entity in a biological sense. It has become part of a larger system that operates on a planetary scale. Thus, this region describes the continuation of death beyond the body. The processes that began with the loss of circulation and cellular control now extend into the environment. Carbon returns to the atmosphere, nitrogen to the soil, water to the hydrological cycle, and minerals to the earth. The transformation is not abrupt but gradual, shaped by conditions and time. It reflects the fundamental principle that matter is conserved and continuously reused within the natural world. At the innermost region of this subject, the scientific view reaches its limit. The body has already passed through visible stillness, failing breath, halted circulation, loss of brain fuel, fading consciousness, final neural activity, cellular crisis, chemical breakdown, cooling, stiffness, self-digestion, microbial decomposition, and return to the environment. What remains is not another deeper tissue or process layer within the body, but the boundary of scientific explanation itself. Science can describe in detail what happens to the organism. It can measure the loss of circulation, the collapse of neural coordination, the breakdown of cells, and the redistribution of matter. It can also define, with increasing precision, the criteria by which death is recognized in medicine. Yet when the question turns from bodily function to subjective experience beyond measurable function, science becomes more restrained. It reaches a point where observations remain strong, but interpretation must narrow. In medicine, death is not defined by a single philosophical idea. It is defined through biological criteria. These criteria differ somewhat by context, but they are built around one central fact. A human organism is alive only while it maintains integrated function. This includes circulation, respiration, brain activity sufficient to support consciousness and bodily regulation, and the coordinated operation of organs and tissues. When these systems cease irreversibly, the organism is dead. Irreversibly is important here. It means the functions will not return on their own and cannot be restored by available medical intervention. This distinguishes death from temporary unconsciousness, deep hypothermia, or certain critical states in which life signs are profoundly reduced but still recoverable. One medical pathway to determining death is the irreversible cessation of circulatory and respiratory function. In ordinary terms, this means the heart has stopped pumping blood and breathing has stopped in a way that will not resume. Another pathway is the irreversible cessation of all functions of the entire brain, including the brainstem. This concept, often called brain death in clinical language, reflects a state in which the brain can no longer support consciousness, spontaneous breathing, or integrated neurological control. Although machines may temporarily move air in and out of the lungs and keep blood circulating, the organism no longer has the brain function required for independent life. These criteria are carefully tested because the diagnosis carries great medical, legal, and ethical weight. Scientific and medical consensus holds that when these functions are irreversibly lost, the person has died. What science can say with high confidence is therefore extensive at the level of the body. It can say that consciousness, as ordinarily understood, depends on organized activity across large networks of the living brain. It can say that this activity requires oxygen, glucose, circulation, and intact cellular function. It can say that when these supports fail beyond recovery, the known conditions for consciousness are lost. It can say that memory, perception, awareness, language, and self-reflection all depend on the integrity of neural tissue and its metabolism. These are not assumptions. They are supported by decades of clinical neurology, brain imaging, electrophysiology, pathology, and intensive care medicine. Science can also say that subjective experience, while deeply real to the one having it, is difficult to study except through behavior, reports, and physiological correlates. This difficulty becomes greater at the edge of death, where reports are rare, timing is uncertain, and direct measurement is limited. Some people revived after cardiac arrest describe experiences during periods near death. These may include vivid imagery, a sense of detachment from the body, unusual calm, or striking clarity. Such reports are important and deserve careful study. They show that the mind under extreme physiological stress can generate powerful experiences. What they do not yet show with certainty is that awareness continues after the brain has irreversibly lost the capacity for organized function. Observations indicate that these experiences occur near the dying process. It remains uncertain exactly when they arise and how they map onto the final measurable activity of the brain. This distinction matters because science works by linking claims to evidence that can be tested and reproduced. If a claim concerns blood flow, brain waves, oxygen levels, or cell injury, it can be investigated directly. If a claim concerns what happens to consciousness after the body has lost all measurable support for mental function, science faces a methodological limit. It cannot easily observe or test a state that leaves no measurable trace and no reliable pathway of access. This does not prove that nothing exists beyond that limit. It means only that science, as a method, cannot currently confirm more than what its instruments and observations allow. The honest scientific answer includes both knowledge and boundary. At present, the strongest conclusion is modest and clear. The personal consciousness known in life depends on the living brain. Damage to specific brain regions alters memory, emotion, attention, identity, and awareness in predictable ways. Loss of whole brain function ends the known biological basis for those capacities. Therefore, from the standpoint of current science, when the brain irreversibly ceases to function as an integrated organ, the measurable basis of the conscious self is gone. This is not a statement about meaning, soul, or metaphysical destiny. It is a statement about what can be inferred from evidence concerning the relationship between brain and mind. Uncertainty remains, but it has a specific shape. It is not uncertainty about whether tissues decay, whether circulation stops, or whether neurons require energy. Those matters are well established. The uncertainty concerns whether subjective experience can persist in any form when the measurable systems associated with it have ended. Scientific observations do not currently provide evidence strong enough to affirm that it does. They also cannot fully close every philosophical possibility. As a result, science neither confirms an existence beyond death, nor treats it as part of established biological knowledge. It remains outside the present reach of empirical conclusion. There is also a broader scientific perspective on continuity that does not involve personal awareness. Matter persists, energy changes form. The atoms that make up the brain and body do not vanish. They enter other systems. Information in the ordinary neural sense, however, depends on living patterns. A memory is not stored as a detached object floating independently of tissue. It is embodied in physical structures and dynamic networks. When those structures are destroyed and those dynamics cease irreversibly, the memory as a living process no longer exists in the same way. This is one reason neuroscience treats the end of the brain's integrated function as the end of the individual biological mind. So the scientific answer, at its deepest point, is both detailed and limited. It states that death is the irreversible breakdown of the organism's integrated functions, culminating in the end of the living brain processes required for consciousness, as science understands it. It states that the body then follows ordinary chemical and ecological pathways. It acknowledges that some experiences may occur near the threshold of death, and that research continues into how the dying brain behaves in its final moments. And it states with equal care that what lies beyond measurable function remains uncertain, not because science has discovered something terrifying, but because science can only speak with confidence where evidence reaches. In this final inner region, nothing dramatic is added to the earlier processes. Instead, the body of evidence is gathered into a boundary. Science explains the dying organism with great depth. It can describe how life ends in tissues, organs, and systems. It can define when death has occurred in medical terms. It can relate mind to brain with powerful evidence. But at the exact point where measurable function ends, the scientific voice becomes quiet and exact. It says what is known. It marks what is not known. It goes no further than evidence allows. The body has become quiet in every measurable way. Movement has ceased, warmth has faded, structure has softened, and the processes that once sustained life have completed their course. In this final passage, the pace slows even further, and the description rests on continuity rather than change. The matter that once formed a living human being has entered the wider systems of the earth, where it will continue to move without urgency or direction. Air passes over what remains, carrying traces of released gases into the open atmosphere. Soil holds what settles into it, distributing nutrients through layers that support unseen networks of roots and microorganisms. Water, whether in the ground or the air, receives what dissolves and carries it onward through cycles that repeat across seasons and landscapes. Nothing moves with intention. Each element follows its own path according to simple physical and chemical principles. Time extends beyond the scale of the human body. The transformations that began in minutes and hours now continue over months and years. Materials that once formed tissue become part of soil composition. Elements move into plants, into other organisms, into the atmosphere, and back again. The identity of the body is no longer preserved in structure, but its substance remains within these cycles. The distinction between what was once separate and what surrounds it becomes less defined. There is no further boundary to cross in the biological sense. The processes described have reached their natural conclusion. What remains is not an event but a state, one that continues without interruption or reversal. The scientific account, having followed each layer from the outer world to the inner structure, now rests at a point where description becomes still. In this stillness, the explanation does not expand. It holds only what has been observed and understood. The body returns to the same systems from which it once drew its substance. The cycle continues without pause, without memory, and without need for direction. The quiet remains complete, and the account comes to rest with it,